Greetings. Welcome to an introduction to statistics for metabolomics. My name is David Wishart. I'm at the University of Alberta in Canada. I am the co-director of the Metabolomics Innovation Centre, or TMIC. This is part of a two-part series on using statistical methods for interpreting and analyzing metabolomic data. This first part will focus on how you can use statistics and how to interpret them in a simple, non-mathematical way. In particular, we have 5 learning objectives for this particular lecture. First is to teach you a little bit about data types that we use in statistics. Second part is to help people understand a little bit more about distributions, statistical distributions, and the measurement of significance. Third part will focus on univariate statistics, the application of student t-tests and analysis of variance, or ANOVA. The fourth part of this lecture will focus on correlation and clustering, how to measure them, how to perform them. And the last part, which is perhaps the most complicated part, is on the aspect of multivariate statistics, the application of principal component analysis and partial least squares discriminant analysis, or PLS-DA, and the application of PLS-DA towards the measurement of receiver operating characteristic curves, or ROC curves. Before going further, perhaps it might be useful to have a few interesting or humorous quotes about statistics. People have many different views of what statistics can do, what it means, and how useful or not useful it is. The early days of statistics started in the 1800s, and they were used in a variety of applications, also in areas of politics and polling. A notable quote from Benjamin Disraeli, who was one of the prime ministers in the UK in the 1800s, was that there are 3 kinds of lies: lies, damned lies, and worst of all, statistics. No doubt he was responding to some bad polling numbers that he got. Another person, unknown to this time, indicated that 98% of all statistics are made up. And given these times that we live in, that's probably truer than we realize. Another comment by Aaron Levinstein is that statistics are like bikinis. What they reveal is suggestive, but what they conceal is vital. This is quite true, particularly in the way that statistics is often interpreted or misinterpreted. My preference in terms of thinking about statistics is that it's a form of mathematics that provides a way of formalizing intuition or impressions. So I often say that statistics is the mathematics of impressions. Many things that we see in life we can interpret and say, that's a big difference, or that's a small difference, or that's significant, or that's not significant. But we don't generally quantify them in a formal way. What statistics does is it formally quantifies or justifies whether something is big or small, or whether the difference is large or small. Now, before going further, I think it's worthwhile outlining or perhaps discussing a little bit more about the type of data that we deal with in statistics. Statistics is fundamental to data science, and data science needs statistics. Both need lots of data, and in this world it's easy to collect a lot of data electronically with all kinds of instruments and devices. What we work with in statistics is either numerical data or categorical data. Numerical data can be either discrete, integer-based, or continuous, which is decimal-based. Categorical data can be either 2-state, binary, nominal or named data, or ordinal data, which is similar to numeric data, but a little different. If we look at numeric data, and we're thinking particularly in the field of metabolomics, genomics, or proteomics, we can think of the continuous decimal numbers like microarray spot intensities, metabolite concentrations, integrated peak areas. Discrete data primarily integer data, could be something like number of transcript reads that you might measure from an RNA-seq experiment. Ultimately, it could be age, or length in meters, but without decimals. Now, numeric data is typically distributed. The number or occurrence of certain classes of numbers can follow a normal distribution or a Gaussian distribution, a Poisson distribution, a random distribution, extreme value distribution. These are characteristics of either the physics, the chemistry, or the biology of the sample that you're analyzing. It can vary a lot, but for most biological systems, the normal or Gaussian distribution is most common. Now, in addition to numeric data, there's also categorical data. Many cases in statistics, we're looking at two categories, and we may measure healthy or sick, treated or untreated, male or female. That's a category where we may collect a whole bunch of numbers associated with those categories. So there's numeric data for the categories, but the categories themselves are binary sets. They can be 0, 1, yes, no, case control, sick, healthy, treated, untreated. That's the most common form that we work with in terms of categories in metabolomics data or other omics data. Other cases, we can look at more categories, nominal data or named data. Typically we use that, and there's 2 or more groups, usually more than 2 groups. They could be social situations or status. In nominal data, there is no order. On the other hand, in ordinal data, there is order, and ordinal data is typically used in things like movie ratings or restaurant ratings. It's often used in things like disease or pain severity ratings or dose and response ratings. So these are the 3 main types of categorical data, and they are often critical in developing and assessing statistical significance or distributions. That allows us to segue into the next major topic in statistics, which is significance and distributions. Before going into that, it's probably worthwhile talking about some other terms that I'll be using throughout this lecture. The first one is to remember the term dimension. We live in a 3-dimensional world, but in statistics, dimensions actually refer to the number of variables. So if you have 100 metabolites, technically you have 100 dimensions. If you have 50 peaks, you have 50 dimensions. The number of dimensions is something that we are usually focused on reducing to make things more understandable. I'll talk about that later when we get into principal component analysis. Another term to remember is univariate, meaning single variable or one variable. With data that we collect on a given population, we are typically in a univariate method measuring a single dimension. So it might be height, or it could be weight, or it could be length per subject. So if we have 100 subjects, we might measure their height. That is a univariate measurement or a univariate data set. Now, if we're measuring more than height, say height and weight and eye color and hair color, those are multiple variables, and so now this becomes a multivariate measurement. So if we have 100 individuals and we've measured 4 variables, height, weight, hair color, eye color, that is now a multivariate data set. In the world of genomics, proteomics, metabolomics, and other omics sciences, most things are multidimensional, multivariate, where we measure hundreds of concentrations of metabolites, thousands of proteins or genes all at once on all of the subjects at once. So in that regard, omics data is very complex and very high dimensional. Now, let's start off with the simplest data set and the simplest approach to statistics, and that's univariate statistics. For this example, we'll just sort of take a population, individuals, and perhaps we'll just focus on height. So there's a collection of about 25 people here, all shown in profile, and we can see some are tall, some are short. If we are focusing on just a single variable, when a population, we are dealing with univariate statistics. Single variable, and in the example I'm giving here, working with, we might be measuring their height, we could be measuring their weight, we could look at their test score, their IQs, but in either case, it's a single variable that we're looking at. If over this population, and 25 is perhaps a little small, let's say we had 250. If we looked at all 250 individuals in that population and measured their height, and measured the frequency at which that given height was recorded, we would end up with something that looks like this. So if the minimum height was 4 feet 8 inches and the maximum height was 6 feet 10 inches, let's say, we would see a distribution where there'd be probably a small number of people at 4 foot 8 and a small number of people at 6 foot 10, and a lot of people hovering around 5 foot 7, 5 foot 8, if we were looking at both men and women. So the frequency of the average height for across genders and populations is around 5 foot 6 or 5 foot 7. We would end up with a peak at that approximate height, where the most frequently measured height is 5 foot 6. This curve, if you look at the profile, looks like the shape of a bell, so it's sometimes called the bell curve. Most frequently it's called the Gaussian curve, which is named after the person who discovered it or described it most thoroughly, Carl Friedrich Gauss, a German mathematician. It's also called the normal distribution because it's what we see in most normal measurements. The equation describing the bell curve is given in the top right. It's a probability curve where P is probability, and it's of the form e to the minus x squared. And you can see that it has some variables. There's the x squared, there's also mu, the Greek letter m for mean, and sigma, the Greek letter for s, for standard deviation. So the bell curve is a function of both the variables x, and its shape and intensity is described by sigma, the standard deviation. The normal distribution, the Gaussian distribution, is inherently a symmetric distribution. It's shaped the same on the right as it is on the left. It has an average or a mean, which is marked with mu, and it has a characteristic width at about half height, which is called the standard deviation. Whether you measure things in biology or in chemistry or in physics, most measurements will tend to follow some kind of Gaussian or normal distribution, where there's some variation about the mean, and that variation can be wide or narrow depending on the phenomena that's being analyzed. So the fact that this distribution is seen in so many forms in nature, and the fact that it's reproduced in so many cases, has made it one of the most important and perhaps the underlying feature to most statistics. What we've also found from many empirical observations is that the more measurements, the more samples, the more individuals or subjects that you include in measuring distribution, the more normal that curve becomes, the more bell-shaped it looks, the smoother the curve looks. Typically, a minimum set to be able to have good statistics and good confidence that there's a normal distribution is about 30 to 40 subjects. And this is just a good rule of thumb. It's also what dictated the typical or preferred size of classrooms in schools. These were driven by statisticians and educators who felt that having that sample size was sufficient to be able to assess students and their progress. So the rule of 30 to 40 is one that still holds, and it's still a useful remembrance or mnemonic about what to use when collecting data for almost any scientific experiment. The Gaussian distribution can be divided into sections or segments that are marked off by standard deviations. So -1 to +1 standard deviation will cover about 68% of the area under the curve. -2 to +2 standard deviations will cover about 95% of the curve. And -3 to +3 standard deviations will essentially cover 99.7% of the area under the curve. So standard deviations allow us to come up with a measure of the width or characteristic width of a Gaussian distribution.